Hey guys, welcome to my channel. This is Omolo the Great. Remember, this is one of the other highly requested videos, and we are going to be talking about a synapse. If you're new to this channel, please remember to subscribe. Hit that button. Please hit that button. Alright, and don't forget to share this video with your friends, family, and colleagues. Alright, so today we are going to talk about the physiology of a synapse, or what we call synaptic physiology. So what is a synapse? And some of the things we're going to look at today. We want to know the definition of a synapse. Okay. We want to know the different types of synapses. We want to know something about synapses have different properties. We want to know the different properties of a synapse. Okay. We want to know about the neuromuscular junction. We shall also talk about the neuromuscular junction. And after knowing about this, we shall also know about the different disorders, different synaptic disorders and disorders of the neuromuscular junction. Alright, so what is a synapse? So a synapse is a physiological continuity between two neurons. Okay? So it is a physiological continuity through two neurons. So the whole of this is one neuron and this is the other neuron. Remember in our previous video we talked about the structure of a neuron, how it functions and how it plays its different roles. And so these two structures make up the synapse. Okay, so we have, importantly, you should know we have a pre-synaptic knob, and then we have a post-synaptic knob. Okay? And then in between here, this is what we call a synaptic cleft. So we're going to expound more on this structure, but this is what you should remember. So a physiological continuity, in that if you have an impulse that is coming right away from your presynaptic knob, this impulse goes through a continuity, or what we call a synapse, or what we call also a junction. So a synapse can be a junction between two neurons, or a physiological continuity through two neurons. So a synapse, and this synapse has to be, information coming has to be transduced and taken through the other end of the neuron. Okay, so what are some of the different types of synapses? So we can classify synapses, their classification. We can classify the different types of synapses in different ways. According to the anatomical classification. So we have an anatomical classification. Okay? Then we also have a functional classification. We have a functional classification. So how do we classify these synapses under anatomical classification? So these can be axodendritic synapses, but they can also be axosomatic synapses, and then we can also have axonic synapses. Axonic synapses. Okay? Whereas the functional synapses can be, you either have a chemical synapse or you have an electrical synapse. Okay, so let's try and put this into perspective. For a dendritic, for the axodendritic synapses, if you remember the structure of our neuron from our previous video, I'll just draw a schematic representation. Okay, like this. Okay, so remember we have we have the axon, the presynaptic knob, the axon, and then we have this, which is the cell body. Okay, this is our nucleus, then the cell body, and then it has different processes. These processes are what we are calling our dendrites. Okay, so this classification, the anatomical classification, is based on this structure, whereby it depends on where is the synapse formed. So if you have an axodendritic synapse, implying that an incoming neuron is synapsing with the dendrite of this neuron. Okay, so this type of synapse is what we are calling an axodendritic synapse. So this is axodendritic because it's what we are forming a synapse between one incoming axon and the dendrite, and then we have another which will be formed between the cell body, or what we also call the soma, and this neuron. Okay, so and then this synapse is what we call the axosomatic synapse. Remember we said the cell body is also called a soma. And then the last classification, we have an axon that is synapsing with another axon. Okay, so an axon synapsing with that can also be this. Okay, so these are some of the examples. So this is typically this and this. These are typically axonic synapses. Okay, so this is the anatomical classification. Literally depends on the two structures between which this synapse is formed. And then for the chemical classification, we are going to expound more on the different on these chemical and electrical synapses. But before I go there, I want to bring to your perspective about the different properties of these synapses. So we will explore about the properties of the synapses. Okay, one, when we look at this first property of a synapse, is that they have a property called the one-way conduction. So synapses move or propagate their impulses in one way. One-way conduction is the first property of synapses. This property is also called the Bell-Magendie law. Bell-Magendie's law. And what does it mean? It means that impulses can only be propagated unidirectionally in one direction. Okay? Unidirectional. In that an incoming impulse that is coming in from this side will only take this very direction, this way. So you won't have an impulse that is coming, it comes and then goes back. But one thing you will remember, when we shall talk about the chemical and electrical synapses, you realize that this law is only specific to chemical synapses because they are unidirectional. So impulses moving in one direction. But you realize that the electrical synapses can actually move bidirectionally. Okay. The second property that we need to know, the second property is that these do what we call summation. So the second property of synapses, that they can undergo summation. Summation, there are two types of summation. One, we have what we call the temporal summation, and then we have what we call a spatial summation. Okay, so temporal summation, let me illustrate using a diagram. You will understand this better. So this is our temporal summation. I'll make it a little bigger for you. So under temporal summation, you're having one presynaptic neuron that is being fired consecutively and is influencing more than one neuron on the other end. So you're having successive stimulations that are coming through one presynaptic node that is stimulated successively to cause effect in the other, at least more than one neuron. This is typically what we are calling the temporal summation. Whereas in the spatial summation, you're having several presynaptic nodes that are being stimulated simultaneously. Okay? Simultaneously to cause an effect. So you're having several stimulations, okay? Different presynaptic nodes that are stimulated simultaneously to cause an effect on the other postsynaptic node. So this is like sort of an addition of the effect of stimulation in different synapses. And this effect can be either through one presynaptic node that is being stimulated successively, okay, that is temporal summation, or several presynaptic knobs that are being stimulated at the same time or simultaneous to cause an effect that is desired on one end. The other property is that they have what we call, they exhibit a synaptic delay. Synapses exhibit a synaptic delay. So what does synaptic delay mean? This is a period between, it's a duration of time that occurs during transmission of an impulse, and it occurs due to certain things that are taking place at the level of the synapse. So this is sort of a delay, a brief delay in synaptic transmission that occurs. So, for example, if we have, this is our incoming neuron, okay? Let me do this. And this is our presynaptic, and this is our postsynaptic neuron. So an incoming impulse, we are going to talk much about how impulses are propagated through. So usually you have an incoming impulse that causes changes to cause release of neurotransmitters. So these neurotransmitters need to move through to be released into the synaptic cleft, and then from the synaptic cleft, then they need to act on the postsynaptic membrane, then to cause the effect of action on the postsynaptic membrane. So all these events cause a delay in the speed, or a delay in the duration of transmission of this impulse. This synaptic delay is usually from around 0.3 to 0.5 seconds. Okay, so and what accounts for this synaptic delay? One, we've said, is the time taken to release the neurotransmitters. That's one delay. Two, time taken for that neurotransmitter to actually move through the synaptic cleft. So the neurotransmitter has to move through the synaptic cleft, okay? And then the third cause of the delay is the time taken for this neurotransmitter to actually cause an effect, or the action is supposed to effect at the level of the postsynaptic node. So the delay also comes during the effect creation by the neurotransmitter. So these three things account for the synaptic delay, which is usually about 0.3 to 0.5 seconds. And so usually the synapse has about 20 to 40 nanometers in diameter. And so that's where most of these processes actually occur. Another most interesting property of these synapses is what we call, it's number 4. Synapses also do what we call fatigue. They undergo fatigue, okay? There are different types of fatigue. When we shall start discussing skeletal muscles, you realize that actually fatigue can be contraction fatigue, can also be transmission fatigue. But typically in these synapses, it's a type of fatigue called a transmission fatigue, okay? So the synapses can fatigue, and typically we're talking about a transmission fatigue. And this fatigue is occurring due to something significant called the depletion of neurotransmitters. So usually consistent stimulation of this presynaptic knob, and constantly keeps releasing these neurotransmitters. A point can be reached when there is a depletion of neurotransmitters. There are no more neurotransmitters required for impulse transmission. And so that is a point called fatigue. So that form of fatigue is called a transmission fatigue. All right. So the other property of synapses is called, anatomically in their structural arrangement, synapses can undergo convergence, but they can also undergo divergence. Okay, so these are other two important. These will come out well when we start neurophysiology. So convergence, this is what it means. You're having several synapses. For example, this is our neuron, okay? So you're having different synapses that are converging at one presynaptic end, okay? At one postsynaptic end. So you're having different of these synapses that are converging onto one postsynaptic end, okay? Whereas a divergence, you're having this information that is being sent to different synapses. Okay, so you see this information that is coming in like this has to diverge to these different synapses, whereas this information has to converge to cause one significant effect. So these are some of very important properties of a synapse, but also there are electrical properties of the synapse, and these ones synapses can be under the electrical properties. One, you can have what we call the generate potentials, called an excitatory postsynaptic potential. Okay, and then two, you have what we call an inhibitory postsynaptic potential. So these are important properties of synapse, whereby under excitatory postsynaptic potential is when the impulse that comes in, okay, is going to cause release of neurotransmitters. Impulse coming in is going to cause release of neurotransmitters, and the effect caused on these the effect the neurotransmitters cause at this postsynaptic neuron is actually generation of a depolarization, or cause a depolarization. So if the effect caused by that incoming impulse is to generate a depolarization, this is what we call an excitatory postsynaptic potential. But if the impulse coming causes release of neurotransmitters that will result into release of ions that will cause a repolarization, okay, this is an inhibitory postsynaptic potential. So put this into perspective, and I'm going to expound more. When you put this into perspective, when the impulse has reached here, causes release of neurotransmitters, these neurotransmitters reach the postsynaptic neuron. So if they cause increased conductance of sodium and calcium ions, the tendency is to cause a depolarization. Okay? Whereas if they cause increased conductance of potassium ions, that means they'll cause a repolarization, which will give us an inhibitory postsynaptic potential. So this summarizes the different properties, the definition, the synapses, and the different properties of these synapses. And so next we are going to discuss something about the neuromuscular junction. All right, so let's look at our neuromuscular junction. The neuromuscular junction. Okay, so the neuromuscular junction is just a connection between one neuron and a muscle layer. Okay, so but its properties and function are not different from a synapse formed between two neurons. So the illustration you see here is typical of the synapse that is formed between two neurons. But also for a neuromuscular junction, it can just be, we have then a muscle layer, okay? It may be skeletal muscle, for example. So this kind of synapse is what we call the neuromuscular junction. We already say the synapse is a junction between two neurons or between two of these nervous connections. So, but the properties and functions are the same, like we've discussed. And also this, I'll use this illustration to make things quite clear. Okay. All right, let's start. So during transmission of an impulse, when the impulse arrives, okay, this is our incoming impulse, there are changes that will occur. We've been talking about the inhibitory, excitatory, postsynaptic potentials. We want to see how is this wave of, how is this impulse going to cause changes in the postsynaptic knob or neuron. Okay, so this impulse here comes, remember an impulse is a wave of depolarization. Okay, so this wave of depolarization is changing the membrane potential. Okay, so remember, this is maybe the membrane was at rest. Okay, so you keep changing the membrane potential. This is maybe plus 40. This may be from negative, for example, 90 or 70. So the membrane potential keeps shifting from more negative to less negative. Okay, I reach the threshold until an impulse is generated or an action potential. So this wave of depolarization is propagated, whereby inside of the membrane is successively made more positive. Okay, so this positivity is channeled down towards this axon terminal. And so what does it cause? When it reaches here, remember the voltage has been changing. There is a voltage that is reached that is going to cause opening of these channels, which we call the voltage-gated calcium channels. So these are voltage-gated calcium channels, these ones, voltage-gated calcium channels. So because of this incoming impulse causes opening of this voltage-gated calcium channels. So we open this voltage-gated calcium channels. When these voltage-gated calcium channels are open, then you're going to have an influx of these calcium ions. Same thing here. Okay, you're going to have an influx of calcium ions. So you have more and more and more calcium that's going to come within this presynaptic node. The role of calcium plays a very fundamental role for neurotransmitter release. If you don't have calcium that is influxing into this node, there will be a challenge in release of these neurotransmitters. So, as we are going to be talking about each as we build the disorders, okay? So if there is a problem with this channel, voltage-gated calcium channel, implying that you don't have calcium that will be influxing into the neuron. And one typical condition that affects this is called the condition 1. This is called Lambert-Eaton syndrome. Okay, so the Lambert-Eaton syndrome, this is a typical autoimmune disorder that is characterized by antibodies that are produced against these channels. So you're having antibodies that are produced and they're destroying this calcium channel, this voltage calcium channel. So when they are destroyed, you will not have, even you have an impulse coming, you will not have influx of calcium ions. That is condition number 1. And so typically it's going to impair the transmission of impulses. So you find someone is really quite slow to react to activities, okay, and they have sort of also muscle paralysis and weakness generally because of the depletion or unable to form these calcium ions within the presynaptic node. Okay, so let's continue. So when this calcium comes and then binds onto these vesicles, okay? When calcium binds onto these vesicles, like you're seeing in blue, okay? I'm magnifying it here. This blue structure here is what we are calling our vesicle, okay? This is a neurotransmitter vesicle, okay? So it contains a lot of neurotransmitters and are usually multiple within our presynaptic node. We already talked about the structure of this in our previous video. I'll attach a link to this video. So when we are talking about this, so what happens? Calcium binds and then causes the translocation of this vesicle, or what we also call an endosome, having a lot of neurotransmitters. It translocates it towards the membrane of the axon terminal, so that these neurotransmitters can bind, okay, to this membrane, and then they can release the neurotransmitters down into the synaptic cleft. So this is what happens. There are some proteins that play a big role in this attachment of this vesicle onto the axon terminal. So let me try and name them. One, you have what we call, this is called synaptobrevin. It's called synaptobrevin. Okay, so this is an important protein. This protein docks onto two other proteins. One, this is called syntaxin. Okay, and then this is another category of proteins called the synapse proteins. Okay, so in absence of any of these proteins, you will not have the ability of this neurotransmitter vesicle to bind and dock onto this axon terminal. This black structure you see here is what we call, it's an active zone. Okay, so on the axon terminal there are different active zones. And what's the role of this active zone? This is the site at which the neurotransmitter vesicle actually binds onto the axon terminal. It does not bind anywhere. It docks onto this axon terminal at the level of a point called the active zone. And it's at that active zone where we have these important proteins that actually receive, or where the synaptic vesicle, where the neurotransmitter vesicle docks onto or attaches onto this membrane. Okay, so these are very, very important. Let me attach the clinical relevance of this. One, you've heard of Clostridium species, the bacteria, Clostridium bacteria. Okay, Clostridium bacteria. Okay, most importantly, one that will affect some of these bacteria, some of these proteins. We have Clostridium botulinum. Okay, botulinum is a very wonderful toxin that's usually used to treat conditions where you have muscles that are stiff. Okay, they try to use botulinum because it will cause some bit of relaxation. But let's see how this happens. So under normal circumstances, this bacteria, when it reaches within this at this level of this nerve tissue, one, has different strains. There are about 7 strains from A up to E. Okay, but the common strains that affect humans, we have strain A, B, and E. So this is what we are interested in. So strain A and E, they usually affect this protein here. Okay, so they come and destroy this protein, and specifically they destroy a specific class of this protein called SNAP25. So they destroy this protein, implying that this synaptic vesicle or this neurotransmitter vesicle cannot attach onto this active zone because of the absence of these SNAP proteins. Okay, but also the other strain, which is B is specific to this synaptobrevin. So Clostridium botulinum, this strain B, will come and destroy the synaptobrevin, implying that if you destroy this interaction, you're going to have challenges with attaching this neurotransmitter onto the axon terminal. And so this causes different presentations, okay? Because you cannot release neurotransmitters, then you're going to get into a state of flaccid paralysis, okay? So the botulinum, Clostridium botulinum, typically will give you a flaccid paralysis, okay? Because you cannot move any neurotransmitter. So the nervous connection or impulses cannot be propagated down into this into the other axon terminal. Okay, so that is typical about botulinum. In our previous video, we also talked about another bacteria called tetanus or Clostridium tetani. Okay, so Clostridium tetani. We said from our previous video, we said it optimizes the retrograde axonal flow. It usually binds onto receptors on the presynaptic knob and then uses the retrograde axonal flow. It moves back up to the level of the cell body, and then now it is reintegrated and channeled down into the presynaptic knobs. When it reaches there, what happens? It is going to prevent the release of neurotransmitters. Specifically, we have glycine and GABA. Okay, these are inhibitory neurotransmitters, gamma aminobutyric acid. These inhibitory neurotransmitters will, when they are absent, implying that when they usually cause an inhibition or to stop the propagation of the impulse. But if they're absent, you're going to have successive stimulations or rapid stimulation that are successive, and you get into a state of tetany, or what we call muscles will be observed in a state of what we call the spastic paralysis. Okay, and the classical presentation of these persons in this form, they get what we call a lockjaw. Okay, a lockjaw. This lockjaw is just typically spastic paralysis of the masseter muscles, these muscles of mastication, the masseter muscles. Okay, so spastic paralysis of these masseter muscles is a typical presentation of these patients with tetanus, and this is what happens. Okay, so let's move back. Let's move back. So with that clinical background, let's see how our impulse is going to move. So the impulse, we said, it causes opening of voltage gate calcium channels, release of calcium ions. They bind onto the synaptic vesicle, giving it a translocation move or push to come and bind onto the active zone where we have important proteins. We have syntaxin, we have the synapse, and doxin with synaptobrevin. So this interaction will cause now release of the neurotransmitters by a process called exocytosis. Okay, So we are going to have our neurotransmitters that are released. Okay, let me just use this to illustrate. So we're having our neurotransmitters released into the synaptic cleft by exocytosis. Okay, so within our synaptic cleft, we already told you this synaptic cleft has in diameter is about 20 to 40 nanometers in diameter. So the integrity of this synapse is actually maintained by two other important proteins that help to attach, so that these neurotransmitters are actually released, so that we don't have an overlap or switching between constant change in the structure of the synapse. So these are kept in shape by two important proteins. We have what we call the neurexin. Okay, we have a neurexin protein and neuroglin. This interaction, this neurexin-neuroglin complex gives the synapse a structural stability at any one point in time. So these neurotransmitters then, when they are released, they come and dock onto their specific receptors. Okay, so these receptors are of different types. You can have what we call nicotinic receptors, you can have muscarinic receptors, you can have metabotropic receptors, but typically in the neuromuscular junction, you have what we call the the nicotinic receptors, which are specific to neurotransmitters that are acetylcholine. So let's focus and use acetylcholine, which is most common neurotransmitter. So acetylcholine that is released binds onto this receptor, which we shall call our nicotinic receptor. Okay, typically the cholinergic receptor, the cholinergic nicotinic receptor. So when this binds, usually causes changes on the postsynaptic neuron. Okay, some of these changes, they're going to result into opening of some channels, and those channels can increase the conductance of certain ions. For example, we can have influx of calcium, influx of sodium. When you have an influx of these positive ions, this changes the membrane potential of this cell from the electronegativity inside to more electropositivity inside the neuron, and this causes an initiation of a wave of positivity or depolarization. Okay, so you're going to have a wave of depolarization that is propagated all through. So this impulse has been pushed or translocated or moved from this side to this side. Okay, so typically when you generate a potential that will result into a depolarization or what we call an excitation, that potential is called an excitatory postsynaptic potential. If this binding of these neurotransmitters result into opening, instead they open other channels, okay, and these channels will cause increased conductance or efflux or potassium ions, implying that they're reducing the electropositivity inside the cell. This will cause the membrane within the cell to become more electronegative, and so this causes a state of repolarization, and this that causes a state of repolarization causes a potential called an inhibitory postsynaptic potential. So these are typical the working of this synapse. The other thing you need to remember is that after this neurotransmitter has done its job, it is actually cleaved. You see this kind of scissor-like structure I've put here. It cleaves these neurotransmitters back into acetyl and choline, which is then channeled back and used for resynthesis of more acetylcholine, and this is called an enzyme, acetylcholinesterase enzyme. Okay, so that's important for us to know, because another condition is what we call myasthenia gravis. Condition 3 called myasthenia gravis. Number 4, myasthenia gravis. This is a kind of rare but common condition that affects synaptic transmission. It's an autoimmune disorder characterized by release of antibodies that will go and destroy specific receptors, and these receptors are these nicotinic receptors or acetylcholine receptors. When they destroy these receptors, implying that no matter how much acetylcholine you release, it does not have receptors to bind. Okay, So the release of these autoimmune conditions results into destruction of these neurotransmitters. So these patients tend to get weakness because irrespective of stimulation, they may not respond to that impulse. Okay, because of the destruction of receptors that are absent to respond to acetylcholine. So that is typical about myasthenia gravis. Okay, so this condition actually can result if you have a tumor of your thymus gland. Remember, the thymus plays a big role in generation of our immunity. If you have the thymus, maybe you have a thymoma, for example, a thymoma that will result into increased priming of the T helper cells that will be formed against these receptors for acetylcholine. So they will come and release, they keep destroying acetylcholine receptors. And that is why as we treat this condition, you can actually do a thymectomy. It has shown to have about 35% remission rate, okay, and a chance of recovery of about 45%. But also you can use their drugs that you may use that may block this the activity of this acetylcholinesterase. And what does it do? These inhibitors of acetylcholinesterase will offer more time or prolong the presence of acetylcholine with this synaptic cleft, so that there is increased chance of the few receptors that may be present on this membrane to respond to acetylcholine. So the longer time you leave acetylcholine there, there's a chance that it actually improve activity. So those acetylcholinesterase inhibitors, those are examples you can use, like pyridoxine. Okay, you can also use neostigmine. These are common drugs that you could use. Okay, so but it's also another important thing, you can offer rest to this person, okay, because to decrease the rate of firing or activity. So when you give rest to this person, offers more time for synthesis of some of these proteins. So I think this summarizes and brings us to the end of our discussion about the synapse, about the neuromuscular junction disorders, the different properties. And please don't forget, remember to subscribe, remember to hit that button and share this video with your friends. We meet next time. Thank you.